Hello and welcome to this webinar titled Do Extractable Protocols Truly Help? An end-user perspective, hosted by BioPharm Asia Magazine and presented by Ekta Mahajan and Dr. Armin Hauk. My name is Sri Patel and I will be the moderator. And before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce you to our presenters. Ekta Mahajan is a Technical Regulatory Program Director at Genentech Roche in South San Francisco, California. Prior to regulatory, she has 15 years of experience in research and manufacturing experience in biotechnology, vaccines, and pharmaceuticals, and lead projects in a wide range of areas, both upstream and downstream, including commercial manufacturing support, process development, and new technology evaluations. Ekta has expertise in disposable technology and led projects for design and implementation of disposables in development and manufacturing, including drug conjugates. She is an active member of BioPhorum Operations Disposables Group, ASTM Working Group, and recently successfully organized the first science-focused single-use international conference through Engineering Conference International in October 2015 in the US and May 2017 EU, which is being converted into series. Before Genentech, she worked as a staff senior engineer in the Technical Operations and Engineering and the Technical Technology departments at Merck. Ekta has a BS in Chemical Engineering from the Thapa Institute of Engineering and Technology in Punjab, India, and an MS in Chemical Engineering from Bucknell University. Dr. Armin Hauk started his career at SIGA in Basel, Switzerland in 1995. He was responsible for the GLP and GMP lab for organic analysis, migration studies, E&L investigations, development of GMP QC methods, and stability testing. After 2010, Armin worked as consultant and qualified person for Intertech Chemicals and Pharmaceuticals in Basel. Since 2016, Armin has a position at Sartorius-Stedim Biotech as lead scientist. Armin is lecturer and trainer in E&L conferences and seminars and is delegate of the German BF arm in the EDQM Pharmacopoeia Expert Group. I would now like to invite our first presenter, Ekta Mahajan, to join our webinar. Welcome, Ekta.
All right, thanks, Shikan. So I'm going to go through, like Shikan mentioned, the famous topic of extractables and how do we really apply at an end user site. So how do these help? A lot of you can go through plastic. It's not something that's new in our world. Plastics came into biomanufacturing, based on what was used in medical devices, used in food industry, and it's part of our day-to-day life, right? We have these water bottles, our condiments come in plastic bottles. So it comes down to what is truly plastic and why do we really care about it? So it's a collection of, the polymer, but also it has stabilizers and fluids, which is helped to, as the plastic is being processed, various aids are added to help with the extrusion process or with the stability and various mechanical properties of the plastic. But why does it matter? One of the disadvantages of doing that is, obviously, it starts seeing extractable and leachables coming out of these plastics. The next question is, since we have been using it for so long, why do we really care? We had it in food industry and medical devices, but the application has changed. Even though it's been in our world for so long, we are now dealing with cell cultures and protein stability. We're dealing with very sensitive cell lines. We're dealing with patient safety, and it becomes critical because it's IV, it's not ingested. When we're dealing with food products, you are ingesting, but with our products, we are, it's IV, so it becomes even more critical. And sometimes these products are used for ocular purposes, which the safety level goes up even more, and a lot of the extractable leachables become even more critical. And then we have to worry about the process suitability. We operate at temperatures anywhere from, at high temperatures, at low temperatures. We store them at low temperatures. We need to worry about the physical characteristics, make sure that nothing leaks, nothing interacts with the protein. And then, last but not least, we need to understand the impact due to changes made by suppliers. Quite a few times when the changes are made, they might have an impact, but and even a small impact can have a huge difference. And I guess we all are aware of the issues that happened, 7-8 years back with the cell culture and the whole ergophos issues. And because the supply chain is so complicated, it becomes even more critical that the suppliers and end users work together to understand the complexity and the impact on our products. So how do we address these challenges? We can continue to work, one-on-one with our suppliers, or we can be more efficient and work as an industry group. Since the challenges faced by all the end users are similar, the products used are similar, so why not work as a cohesive group since we're all working towards same problem or same solution, I should say. So that's where BPOG comes in. The BioPhorum Operations Group is an industry forum that has pretty much all the major companies, and this is an outdated data, but right now, there are even more companies that are part of BPOG, and all the big, small companies are part of this, which makes it great because everyone brings in different experience as well as learnings as they interact with different health authorities or they've had different products. So it doesn't stay just as one company's problem or the interactions of like a sensitive cell line, but when you start seeing scenarios across the industry, you know that it's more widespread versus just for one. So that's where the BPOG protocol came in, and the first initiative that this group took on was looking at what's the biggest problem that we face as we try to implement single-use. And extractable was one of those biggest issues that came up, and the fact that there is no standard protocol as you get, you start working with suppliers, there was different reports, different formats, different conditions that were used, and the conditions that were required to implement at your company were different. So that's where the first goal was to develop a standard extractable protocol for testing single-use components. And the benefits were that it would reduce the qualification time for single-use components from 1 year to 4 to 8 weeks. Because if you were to do yourself, then you have to work on agreements with lab, the protocol report. By the time you do all that, sometimes the need for implementing goes away. But if you have a report from the supplier that meets all the requirements, then it's more about validating internally, making sure it meets the requirements, and then going through the business process. So the qualification time reduces considerably. And this extractable protocol, because it was the consensus of all the companies in the industry, it meets the need of the majority of the biopharma, and each company doesn't have to generate their own protocol, or the suppliers don't have to work with each company to try and generate a different protocol that meets different companies' needs. Once it's done, it's known because it's a consensus protocol, so it would meet everyone's needs. And when the data is available, it helps for troubleshooting or resolution of issues. You don't have to start from scratch. You already have data that you can start using to resolve any problems. And then a lot of people think about, well, you need only leachable studies, but the reality is that you need extractable data to be able to perform leachable studies, and those are a must for stability program and for establishing product expiration. So how did this evolve? So there was a paper that was published by myself and two other co-authors, Krishnari Chaudhary and James Vogel, and this protocol became the basis for, the paper became the basis for the protocol when the BPOG group came together in April 2013. There was a survey that was done. So it was not just something that, the group started working on, but there was a survey done within the companies for these, 24, 25 member companies to see what are the conditions that are required in each company, what are the process requirements, and based on that, the conditions were selected. There were the protocol was generated after that with the consensus of all the BPOG members. This protocol was shared with the BPSA supply group, as well as we had a lot of supplier one-on-one calls to ensure that the protocol met their needs, and there was feedback from the supplier. So the protocol changed, and the current protocol that is out there is actually a reflection of the discussions that we had with the suppliers, and the feedback was incorporated. And then the final paper was then published in November 2014. The protocol was shared with ASTM as well as USP. There are ongoing discussions to make sure that whatever comes out of USP is aligned with the BPOG protocol. It has been incorporated into different companies, and speaking for Genentech Roche, it's been incorporated into our global guidance document as well as for our internal global SOPs. That's what we share with the suppliers and what is required for us to be able to qualify any incoming single-use material. And just a brief snapshot of what the extractable conditions look like, and it focuses on all the single-use components, not just bags, but tubing, connectors. A lot of times people ask about connectors, that they are really small components. Why do you need such extensive studies for connectors? But in reality, as we get more and more into single-use manifolds, and you're getting into half-inch, 1-inch size connectors, and the number of aseptic connectors that can be in a manifold, when you start adding the area, it can actually be, it starts becoming very considerable and almost equivalent to a tubing. That's where the extractable data for connectors start becoming really important. And then there were conditions going all the way from time 0 to 70 days. And again, these are based on the requirements, and time 0 is critical because you need a control, but also a lot of the volatiles come out initially because of the gamma radiation. And in the past, when data was provided only for 70 days or 90 days, depending on different suppliers, we lost a lot of data, the initial data points. So we didn't really see any volatiles or non-volatiles, and semi-volatiles. The only data you could see is non-volatiles. And then also we lost the trending. So a lot of times when the volatiles are coming out, or if they don't go down, but they start forming byproducts. So that's where the range of the time points definitely helps. And then what do we do with this data? I think that's a question that comes up quite a lot. So for the end users, we actually have to perform tox studies. We have to look at what is the estimated daily intake. And in doing that, we look at, and this is just an example. For example, if there was a bag that we needed to calculate the safety profile for, so we look at the surface area of the bag, how many bag ports are there, and sometimes just to be able to qualify multiple bags, we would take worst case. Instead of even if there are 2 or 3 bag ports, we might go to an extended number of bag ports to make sure that we'll be able to use it for multiple scenarios or bag manifold designs. We look at the final product. What is the highest expected dosing for that final product, what is the final grams for that product, and then the typical patient weight of 50 kgs, and then we do a estimated daily intake. Once we perform this calculation, then it is compared to the daily estimate is then compared to what is the safety limit for that particular component or that particular extractable, and if it is below that safety limit, then it's okay to use, and if not, then we cannot use that bag or tubing or whatever it is. So now I wanted to go a little bit into the case study. How do we actually see the value of the data that we got or we've been getting now with the BPOG protocol? So the first one was qualifying for use and have these within 1 month. And that's a very typical request that we get internally, because a lot of times the applications are very urgent, or something, instantaneously doesn't work, or something goes wrong, and all of a sudden it's like, oh, we need to use single use, but you have only 1 month to do it. So that's where, if we don't have the data, it becomes very difficult for us. But if you have the data, then we are able to qualify and use it. So in this case, because it was using organic solvents and other buffers, so the first thing to do was to check if the solvents fall within the model solvents that are listed in our BPOG protocol. The next was, does the supplier have supporting extractable data? So in this case, we were fortunate that the supplier had started doing the BPOG testing, and we had the report from them. So we were able to perform the tox calculation using the method that I showed earlier. So in this case, we did not see any class 1 solvents as per the Q3C. There were 2 class 2 solvents that were detected when they were exposed to the 50% ethanol and 1% polysulfate AT, and 1 class 2 solvent was detected in the back port. But the permitted daily exposure as per the ICH guidelines were below the PD for all these 3 extractants. So we were able to say that it's safe to use. And then we also looked at the summation of all the known extractables and performed similar calculations for the EDI and as well as for the unknown extractables, because so since with the BPOG protocol, there's a lot of new extractants that are being observed. I think the suppliers are still working on identifying those. So in this case, we had to assume that the unknown extractables were toxic, and we had to look at the threshold of the toxicological concern. But fortunately, all these unknown extract summation was still below the TTC level. And then in this case, because we were using 20% DMSO, we also looked at the leachables from if they were to expose to 20% DMSO and performed a similar tox calculation. Based on then all this data, we were able to say that the film is suitable for use, and the project was able to use the film. But in conclusion, for this study, availability of data supported the project timelines. If we did not have data, there was no way that we would have been able to support the 1 month timeline. And the extractables were within the acceptable limit, and the film was suitable for immediate use, and they were able to use the film. So it was a good success story, which doesn't always happen. So the case study 2 is another one of the success stories where we did have the data. There was a leachable assessment that was required to be done, and we needed to do it for bags along with assemblies in 2 months. As you can probably see the trend, the time that we are given is not that long. In this case, the components included the connectors, the bags, tubing, and the leachable. Like I said, the leachable assessment requires extractable data. And again, if we didn't have the data, this would have been an issue. But in this case, again, we were fortunate that the extractable data from the suppliers enabled us to execute the leachable studies to meet the project timelines. One of the and the leachables were within acceptable limit, so we were able to support that project. But if it did not have the appropriate data from the supplier, it would have required 1 year, which meant that the group would have used stainless steel instead of single use, or the project would have been delayed. And we typically don't like delaying the project. So in this case, we probably would have gone ahead with stainless steel, which would have been bad. And that is one of the biggest, the benefits of having the protocol. Now we can start actually implementing single use instead of constantly losing the power because due to lack of data. So now the third case study, which was not a very successful one, or shows the impact of not having good data, is we needed to perform an extractable assessment for a filling line, and the goal was to look to enable the filling of the products in vials using disposable assemblies. Unfortunately, in this case, we didn't have good data from the supplier. It included only TOC results, which makes it impossible to perform any kind of tox calculations. So the outcome was that we had to do internal studies to address the missing information. It took us 6 months to perform the assessment due to lack of data. It impacted the projects. Resources were wasted in deciphering batch supply data. It introduces hurdles for implementation of single use, and as would be expected, the supplier does not stay on the preferred list anymore. So to conclude, availability of appropriate extractable data enables execution of projects, especially the ones with short timelines. Suppliers providing data become preferred suppliers. As it lowers our assessment time, we can focus more on projects, technology projects, or drugs. That is our main business, and efficient utilization of resources. So with that, I would like to acknowledge a lot of people that have been responsible for the success of being able to use the data internally, which was my previous boss, the team that worked on the drug conjugate for this first case study. Krishna has brings a lot of expertise, and then of course the BPOG A&L team. This would not have been possible without that team. With that, I'd like to close the presentation and give it back to Shrikant.
Thank you very much, Ekta. So just as a reminder to our listeners, if you have any questions for Ekta or for Armin, then please click on the questions tab and send them to us. We'll be asking these at the end of the presentation. So I would now like to invite the presenter for the second half of this presentation, Dr. Armin Hauk, to present an industry perspective. Welcome, Dr. Hauk.
Welcome, everybody. Thanks, Shrikant, for the nice introduction. I will now give you a short overview about the aspects which go beyond standardization, which were mentioned before, and I would like to answer the question, do extractable protocols truly help? And my talk will consist of 2 or let's say 3 elements. First of all is a kind of introduction to the topic, the need of extractables data, then some words regarding the standardized extractable methods, then some words regarding experiences with standardized methods we have here at Sartorius. Then I would like to talk about the aspects which go beyond standardization. For that purpose, I need a short excursus into physics. Then I would like to present 2 case studies, one on the use of extractables data and the other on the fate of leachables concept. And then I come to a conclusion and outlook. We have heard in the previous talk that extractable studies are conducted to evaluate the potential patient risk or product quality risk, which are resulting from compounds which may be released from contact materials. And in this context, extractable studies shall elucidate and semi-quantitate any potential releasable compounds associated with the contact materials. So extractable studies are related to the contact materials. And the aim of the leachable study is to quantify drug impurities in the end, which are associated with the contact material. It is clear that extractable studies are valuable in case they allow a reasonable prediction of potential leachables. This was also mentioned by Ekta before. To realize a tox assessment, Ekta showed an example. To plan leachable studies, also here Ekta showed an example. Or to have good reasons to avoid detailed leachable studies. So reasonable quantitative extractable data are obviously required. In the previous talk, the need for extractable data for single-use system and single-use technology obtained with standardized methods was discussed. And we from Sartorius-Stedim, as supplier of such devices, understand that. And it is evident for us that the initiatives from BPSA, BPOG, ASTM, or USP to provide standardized methods are generally highly appreciated by our side, because standardization is the tool to overcome divergent and changing requirements. And we have also understood that extractable data obtained with standardized methods may be beneficial for the biopharmaceutical industry, for material device selection, and for their process qualification. And we have also understood that these data may be used for comparison of materials and can therefore be driver for future developments. Therefore, yes, indeed, standardized extractables protocols can truly help. Also, there are still some open points which are sometimes controversially discussed between the different stakeholders. First of all, the protocols were not developed for our needs, for the supplier needs, and any application outside the biopharmaceutical industry, as classical pharma applications and also ADC manufacturing cases, are not included, and this results in additional tests. The protocols, whether they are from BPSA, BPOG, ASTM, USP, claim to be standardized, but unfortunately, they are not standardized because they are not aligned in extraction solvent and time points. Therefore, for us, this undesired divergence in the different methods still exists. There were some rumors that the proposed solvents cannot be subjected to suitable analytical methods. For example, this was shown in the USP workshop 2 years ago. But last but not least, I would also raise the question whether the method we are using are really optimized, whether they are validated, and whether we know their capability. For example, these methods were never checked in round-robin tests, which are, from my point of view, a prerequisite before these methods become a real standard. I hope that as soon as the USP 665 will come into force, these problems will disappear, because USP will be mandatory, so everybody has to do that. A USP chapter will be per se will show methods which are per se validated, and therefore I think USP 665 will most probably become the standard for extractable testing of single-use system and single-use devices. Now some words regarding the experience at Sartorius with extractables protocols. I would like to highlight here that Sartorius has a very deep knowledge about extractables since more than 20 years. We have a huge number of extractables data available in our extractable guides for our different devices. These are analysis which is done with modern state-of-the-art techniques. We have published on extractables for filters and bag materials in scientific journals, and we have recently conducted more than 20 extractable studies according to a new Sartorius protocol, which covers the needs of Sartorius as manufacturer and which covers the needs of all of our customers. In addition, we have conducted extractable studies based on the BPOG proposal with all 6 extraction solvents and with analog to the USP draft with 3 extraction solvents. And so far we have tested a huge data set for all different tests for 3 filters, 4 housings, and 2 films. So in the end, I can say we have a huge data set for comparison. And just a brief overview to the Sartorius extractables protocol, which is a quite compressed extraction matrix, but followed by subsequently a comprehensive set of analytical tools which are applied from HPLC, LCMS, GCMS, Headspace, GCMS, ICP-MS, etc., etc. And when I now compare results from the different protocols, and here I have an example where I compare BPOG results with Sartorius results for a gamma irradiated sterile filter, then I will find the following. The overall qualitative results of the extracts are nearly identical. We see that the ethanol extract reveals some more membrane-related compounds compared to the BPOG extraction media. We see that water ethanol show in general lower concentration compared to ethanol alone. We see equivalent extractables profile for water and sodium chloride solutions, and we see equivalent extractables profile in any of the acidic or alkaline extractions, whatever protocol we are using. So we can say there are no surprises. All the data we found were as expected. We found partially equivalent results whether we extract with ethanol, water, or polysorbate extractions. This is good to know for the future if we would one day decide to skip the polysorbate extraction. We had one big difference. This is the Headspace GCMS analysis, where indeed the polysorbate extract reveals a higher load of volatile organic compounds in comparison to a water extract alone. Also an interesting difference, good to know. And we found equivalent extractables profile in the elemental analysis. All in all, we can say that the results are more or less comparable, whether we use our protocol or other protocols. And so in principle, I would say this conflict, which is sometimes discussed about the correct extraction methods between the different stakeholders, is from my point of view not worth to be carried on. I would now come to aspects which go beyond standardization and things which have to be considered. And this has to do with two major problems, more or less, which persist even after standardization. This is a question: how can we obtain extractables data for devices of different sizes or for device combinations? And how can we use extractables data in a risk assessment, or how can we extrapolate extractables data to reasonably predict leachables? So the question is scaling and combination of extractables data, and the question is the use of extractables data and extrapolation to leachables. Both questions will be discussed now, and I will make some proposals that we can overcome these so far unsolved problems. But for that, I need a short excursion into physics. When we are carrying out extractable studies, or we are investigating leachables, then we are in a situation that we bring two phases in contact, which are not in equilibrium. And then during the time of extraction, there is a migration of compound from one phase into the other until an equilibrium is reached. This migration can be described by a phase transfer model, a migration model with two adjacent compartments. And here you see a presentation of a plastic layer on the right side, on the left side, and the blue line is the diffusion gradient inside the plastic. And at the interface, you have a concentration jump related to the equilibrium conditions at the interface. For such a model, you can find in the literature very interesting solutions, how you can calculate extractables based on these phenomena. If you are doing extractable studies in a kinetic mode, then you will find curves like that. You get a time-dependent release of extractables or leachables and a temperature-dependent release of extractables or leachables. The first part of such a kinetic can be described with diffusion mathematics, and the second part can be described with equilibrium conditions. The first part can also be described, the temperature dependence of the first part of the curve can be described with the Arrhenius concept, and the second with the Van 't Hoff equation. But all in all, you can say extraction curves like that can be calculated. Why is this necessary for us as suppliers? The problem is that we have to handle numerous materials and devices of different sizes. That means for us, for example, for Sartorius, we talk about thousands of items and hundreds of assemblies. And the fair number we are discussing about is 5,000 items. And you can imagine it's technically impossible to create comprehensive and realistic extractables data for each and every device. The models which are discussed so far to make scaling or to combine different extractables data are, of course, sometimes wrong or misleading. For example, Gengi and Rabinov showed in a presentation which was published in the PDA journal last year that this intuitive approach, Ekta has also shown before, to scale extractables data just via the contact surface is questionable and can violate the physical principles behind. Scaling via contact surface is from a physical point of view only allowed in cases with short contact times where the extraction is diffusion controlled. All other cases need scaling based on equilibrium data. And avoiding hundreds of new measurements requires, in addition, a heuristic approach. With other words, we have to use our data as they are today. And the equilibrium approach is in general reasonable, as the equilibrium concentration is the maximum concentration you can reach in a given system. Let's make a model. Let's make a case study one. Let's make some assumptions. We would use a 3D bag like that here given on this slide, and this is used for a buffer solution, which is prepared in the first bag and is then transferred to the second, to the third, and the fourth bag. And let's assume that this refilling from one bag into the other is conservative. That means a full volume transfer, not providing any new leachables or remove any leachables. Then the question is, what is the right concentration at the end? A pure cumulative worst-case assessment would simply multiply the extractables data with the surface area, in this case with a factor of 4. A correct worst-case estimation, a physically correct worst-case estimation would consider that after each successive storage of the buffer, an equilibrium is reached, which is the maximum achievable concentration. But what is the result then? Assuming that situation, then you can, I made here a model calculation where I assume that this successive refilling of this buffer solution is done. And here I assume that I have a water-ethanol mixture 1:1, and then in the first bag it was stored until equilibrium was more or less reached. Then using the equation given from Gengi and Rabinov, then you can calculate the amount of expected extractables in the four extraction experiments. For a compound which is good good soluble in this solution, you will indeed get a rising concentration. For compounds which are not as good dissoluble in this solution, with higher KPL values, then you can see that you are converging against a certain concentration, which is far from a factor of 4. By the way, I have to apologize. I do not know why the equations here are completely destroyed in this plot. But what is given here is the equation of the Gengi and Rabinov publication, and the other values here are the KPL values, which are the partition coefficient between plastic and the liquid phase. But let's now come to the question number 2. What are we doing with extractables data? Today, when we talk about leachables, then we are more or less trapped in this extractables assessment. Leachables are thoroughly regarded as compounds released from the polymeric material into the process liquid, and the common understanding is single-use systems are sources of leachables. And when you make extrapolations from extractables data to leachables data, then the resulting expectation is there should be a high load of leachables in the products. But if we are doing validation studies on the product level, then we realize very soon there are no process leachables. Leachables do very often not contribute significantly to final drug impurities. So obviously, with our perception, something is wrong. And in this context, Jessica Shia gave a very nice presentation at the RAPRA conference about 2 years ago, where she could show that indeed, in a bioreactor, leachables concentrations are rising the leachables are accumulating, but as soon as you start with the downstream processing, the leachables are disappearing. So obviously, there are very efficient sinks of leachables in our downstream processes. And now we can start thinking or anticipating, what are these sinks? Where are these sinks? And we can also think about, what do we already know? Then of course we can imagine that the cells and the cell debris which is removed in the harvest step may absorb leachables. Of course, cell surfaces can absorb leachables, and when the cells are removed, then also the leachables are removed. We know that sterile filters, the membranes, are very efficient scavenger of leachables. We know that purification membranes are very efficient scavenger of leachables. We know that rinsing of a protein A column can also remove leachables. We know that ultrafiltration, diafiltration in a cross-flow device can very efficiently remove leachables. And there were several publications, for example, from Mahajan regarding a cross-flow. We have a scavenger, the scavenger effect of membranes was shown by me in the conference in Portugal, and the removal of leachables with cell debris will be shown in a publication which will be published soon. So in the end, that means that, but what does it mean for example, for certain devices we are using? For example, what does this really mean for a qualification of a bioreactor when we talk only about the extractables and do not think about the sinks of extractables? What does it mean for a protein A column? We only talk about the extractables from the column material, but what is about the sinks? And the same is true for the membranes and more valid for cross-flow systems. So what I think is, there is a proper balancing necessary where we consider the sources. There are for sure sources, this is clear, but we also have to consider the sinks of leachables. And doing that, we can of course also use quantitative concepts for that, and this is the fate of leachables concept. We can consider that there are sources of leachables. We can consider there is a distribution of leachables, and we can consider there are sinks of leachables, and combine it into one model, which is called the fate of leachables model. And this fate of leachables model and the principal approach there can be very easily applied to our biopharmaceutical processes, because a biopharmaceutical process is a well-defined system of components and devices in subsequent process steps. The process steps are dynamic, but they carried out in closed systems. The boundary conditions for all the process steps are very well known. That means, for example, process volumes, composition, mass flow, temperature, etc. So these are the ideal prerequisites to apply mathematical models, for example, dynamic box models, to model the amount of leachables in such a process, because the different process steps can be described as compartments. In each compartment we can describe sources, distribution and sinks of leachables, and we can calculate it based on physical chemical mechanism. We can apply a strict mass balance conditions between the different compartments, and we can of course calculate exchange between the compartments or the discharge simply with the flow of the liquid phase. And I will show you now a very simple calculation I have made with a bioreactor, with a genetic bioprocess, which consists only in 6 elements: a bioreactor, a harvest step, filtration steps, purification steps, ultrafiltration, fill and finish. And for all these boxes, I calculate the release of leachables with a diffusion model. I calculate the adsorption on the materials based on adsorption isotherms. I calculate dilution and concentration, for example, in the cross-flow step. As input data, I need some data about the process, which are total volume, total mass, thickness of the material, leachables load of the material, biomass, etc., etc. And I need some input data for the leachables, for the compound. And doing such a calculation, I get results like that. What you can see here is a bioprocess which starts with a medium preparation, goes through the bioreactor, go over the harvest step and the downstream processing. And this graph has to be read like that. The red bar is the leachables always coming from the previous step entering the compartment. The green bar is the leachables increase in the process step. and the blue bar is the leachables removal in the process step. And you can see that during the media preparation and during the residence time of the solution in the bioreactor, indeed you have an accumulation of the leachables in the solution. But in the harvest step, due to the fact that leachables are absorbed at the cell debris, most of the leachables are removed. After the handling after the harvest step would of course yield a certain amount of leachables, which is then carried through the process, which is then of course removed by certain filtration steps or by ultrafiltration diafiltration step. This model calculation here was done for a leachables compound A, which is comparable to compounds like deterged butylphenols, typically additive degradation products. And the parameter I have used are given in the gray box here for diffusion constant, for partition coefficient, for ultrafiltration diafiltration factor. You can make a similar calculation, and then you get, of course, a result which is a very low amount of leachables compared to the high amount of leachables which were present in the bioreactor at the end of the residence time, dwell time in the bioreactor. Doing a similar calculation for leachables B, which is a much more better soluble compound, you will see again a rising concentration in the bioreactor, a low absorption on the cell debris, so there's only a low removal of leachables with the cell debris, also a low interaction with the filtration membrane, but in this case, of course, we have to assume that the ultrafiltration diafiltration step will remove these leachables to a much more higher amount because it can be removed with the permeate. And again, you will find in the end a very low concentration of leachables in this calculation compared to the amount which was present in the bioreactor at the beginning. And if you would now compare these plots with the findings of Jessica Shea, I showed some slides before, then you see that this model calculation reflects the empirical findings much better than any worst case assumptions which you can make based on surface areas or things like that. So coming now to a conclusion and outlook, do extractables protocols truly help? My answer was yes, of course. Standardized protocols for extractables testing are indeed valuable because we can avoid a lot of divergent interests in extractable studies. On the other hand, and further I have discussed that the difference in the protocols are in principle of minor relevance. The overall outcome of a comparison of the studies conducted based on the protocols are more or less quite equivalent. But I have also tried to discuss that there are certain aspects beyond standardization which need to be included into our considerations in the future, or which have to be included into our standardized protocols. This has to do with scaling of extractables data, and this has to do with the use of extractables data, in particular the extrapolation of extractables data to leachables. And in risk assessment for leachables, beside only considering sources, what we are doing now, we really have to consider also the sinks of leachables in the process to get better prediction of leachables in a process. And I would finish with a motto from Albert Einstein saying everything should be made as simple as possible, but not simpler. And I think this motto fits very well to our extractables discussion because we have certain things which we handle too complicated. This is, for example, the discussion about the extractables protocols. And there are cases which we handle too simple. And this is, for example, the extrapolation of extractables data to leachables, or this is the scaling as well. At the end of my presentation, I would like to make an acknowledgement to my colleagues from Sartorius-Stedim who contributed directly to this work, which is Ina Pai, Roberto Menzel, and Samuel Dorre, and our lab teams. But I would also like to thank colleagues from other companies for their discussion, for sharing data, graphs, plots for the presentation, which is Jessica Shea from Millipore, Kate Lee from Genentech, and Michael Jahn from Lonza. And then I'm at the end of my talk, and I'm open for question and discussion. Thanks.
Thank you very much, Dr. Hauk. So at this point, I would like to begin the question and answer session. Just as a reminder to the audience, if you've got any questions for our presenters, then please send them to us via the questions tab at the bottom of your screen. So our first question. So Armin mentioned that round-robin tests would be required before establishing a method as standard. Were there no attempts so far to compare results from different labs and devices?
Yeah, and a very interesting question, and this is the reason why I've mentioned that. To my experience and to my knowledge, there were so far no attempts to do that. I would highly appreciate if we could do that, because I am from a GMP laboratory, and to be honest, normally we have methods which are optimized and validated on one hand, and in the technical area we have these round-robin tests to make methods to real standardized methods. So I would really motivate whenever possible to come to a position where we compare our different methods, where we make a round-robin tests with the different methods to get a better picture about the comparability of different results, about the robustness of the methods, robustness of the results. I think this would be highly appreciated, yes.
Great. Next question is for Ekta, although I mean, feel free to add to this. It's quite an open question. Which is preferable, USP versus BPOG?
Well, from the end users, BPOG protocol is what applies to us. USP is a very open-ended protocol. It's not a standard yet, but it applies to large and small molecules. So it's not focused just on biotech, biopharma industry, and that's why the conditions are different than what the BPOG protocol is stating, because the BPOG protocol is very much focused on biopharma industry, and the USP is also very much coming from the drug final container closure, versus the BPOG protocol is looking at the drug substance drug products, like the entire game of. So the work that is going on in discussions with USP to ensure that even if their test points or the solvencies are different, if a supplier has done testing with using the BPOG solvent, they would not need to repeat what is stated in the USP. For example, if the pH 13, that should be able to cover a pH 10 solution. So from the industry, from the end user perspective, we would need the BPOG data. Even if a supplier does USP, we would still require the BPOG.
Great. Next question. So, Armin, you explained how scaling of extractable results can be done with physical methods and how a fated leachables model can look like. But what can we do with the unknown compounds which occur in almost all extractable experiments?
Mm-hmm. Yeah, this is also a very interesting question. The thing is, of course, in principle, you can also include unknowns, and you can also quantitate unknowns. You have to simply give the unknowns physical properties. That means if you have any hint about the physical properties which have to be applied to these unknowns, then of course you can make this calculation and get a quantitative result of an unknown compound. But now the next question would be, where can you get these physical properties of an unknown compound? For example, if you think about things like a partition coefficients which are required for my calculation can be obtained, for example, for HPLC correlation experiments. So in principle, you can clearly say if you have two compounds with a known retention time and a compound in between, then you have good reasons to say the compound which you are talking about has a similar partition coefficient compared to that compound with a similar retention time. So you would have the possibility with this method even to make calculations for unknown compounds.
Great, thank you. Next question. If data with water and PS80 or another solvent is the same, do they still need to perform testing with all solvents listed in BPOG protocol?
Yes. So I think a lot of times those studies are done, or there are questions about the relevability of different solvents. One thing to realize is when the standard was created, standard is supposed to, apply to everything. So even if one company's one component, a bag or a filter or something, shows similar data, that does not apply to all the components for all the suppliers. So for creating a standard, you have to go with the norm that it should meet 80% of the needs, or it would apply to, so different suppliers will have different experiences, but from the end user perspective, we have to go with what we use, how we are able to justify our approach to health authorities. And now being in that group, I'm going to be even more vigilant about this since it becomes even more critical on how we justify our approaches. One of the things that has been done in different medical devices, even like for ocular products and all, is showing that there isn't, the main goal is to show that whatever is, either something is not coming out, or if it's coming out, it is still within the safe limit. So having the data that shows that nothing is coming out if we store something in polysorbate 80, or it's similar when we're storing it in different solutions, actually helps making our case to the health authorities, because then you can show, oh, I can store these solutions, and I know based on the data that nothing is coming out. So to me, from an end user perspective, it's again about the mindset on how we're looking at the data. But for me, those are actually great things to be able to see if we don't see a lot of extractables, or if there's a similar profile from different solutions, that will help us build more and more cases, or easier cases for implementation of single use.
Great, thank you very much. Next question is quite open, so feel free to come with an answer. What is your view in terms of calculating filling needle extractable into vials?
Is this a question for me? Maybe.
It hasn't actually been directed to either of you, so feel free if you've got an answer for that or not.
It's because there is calculating. The thing is, of course, yes, in principle, you can also imagine that you can calculate how a filling needle will release extractables into a vial. This is a dynamic process. That means you need, of course, some knowledge about the diffusion constant of the material in the filling needle. You need the surface, you need the flow through the needle, and then you are able to calculate the amount of leachables which may be released from a filling needle. This is in principle a thermodynamic, a kinetic experiment which can be modeled with good physics without any problems. Of course, you need certain input data. Input data are, for example, diffusion constants. They could be obtained from the literature. You need partition data. Some of them can also be obtained from the literature and can be taken from the extractables experiment after proper evaluation. So in principle, I don't see why we should not be able to calculate leachables which maybe migrate out of a filling needle.
Great, thank you very much. Next one's open as well, so feel free to come in with an answer. How would you be able to calculate all leachables if we don't have all the migration coefficient values?
Yeah, this is also something for me. I think, yes, of course, it is. We have this data gap, for sure. We have a data gap that we do not have for all the leachables, all the data immediately on the table. So we have, for example, not all the diffusion constants for all the materials involved, and we do not have all the partition coefficients which are required. But on the other hand, there are very, very good correlations published from the food contact industry regarding the determination of diffusion constants in different materials. We can make use of that, and the same is true for partition coefficients. So in principle, we can make use of the work which was already done in the food contact area regarding the modeling of the release of leachables from certain materials, where they have also required diffusion constants, partition constants, partition coefficients, and things like that, and also temperature dependence of migration is described there. So, right, there are some data gaps, but of course we can rely on data which are already there. There are good correlations, and I'm quite happy to see that we in near future, we will have all these data that we can really make these calculations.
Great. Next question. How reliable are quantitative or semi-quantitative results with regards to alteration of the material or respective changing of antioxidant concentration, ongoing degradation, etc., during aging and storage of the SUS?
Yeah, maybe this is also something I have to answer, because this goes a little bit in the direction validation, round-robin test, etc. It is true. Today we have a little bit the problem that nobody really knows exactly what happens if you challenge these methods. For example, if somebody alters the additive package, will we really see that in our standardized extractables protocols? If somebody is changing something with the gamma radiation, for example, do we really know exactly the impact on that on our analytics? So there are certain question marks which have exactly to do with that need for optimization, validation, and also this exchange of data. Finally, last but not least, also with a round-robin test to get more information about the reliability, the robustness of the method.
And I would just like to add a little bit. I think I agree with Armin that there is a lot of unknowns right now, and I think it'd be great if someone was to do those studies. But just based on the experience that we have so far and looking at data, there is an impact on with the aging or the storage of the single use, and that was one of the reasons why in the protocol the requirement was to test within 5 weeks of gamma radiation, because if you let it age over time, there is a loss of the volatiles, and the data that you would generate from something that had been stored for some time, or even if you age it at a higher temperature, you would start losing those data points. So there is a definite impact on the quality of the data that you would get if you were to, depending on the radiation dose or the time or the aging of the single use.
Yeah, I fully agree that there are definitely these differences, and maybe hopefully we can see that with our protocols. For example, what we also have not really in depth investigated now is how is analytical uncertainty related to a lot-to-lot variability. To be honest, we have some ideas about that, but in the E&L community, this topic was not discussed so far. And this is also something which needs definitely some more investigation in the future, that we get a clear picture how good are our analytical methods with regards to the lot-to-lot variability. Are we really able to see lot-to-lot variability, or is our analytics so, I would say, non-robust, so non-selective that we cannot distinguish between those?
Great, thank you very much. So the next question, has the BPSA formally published a protocol?
I don't believe so. I think there was a follow-up paper to the BPOG, but I don't think it's a protocol.
Okay. Thanks for that. And next question for Armin. Would you be able to share the leachable concentration predictive model?
Yes, in principle, yes. I could invite the person who wrote this question. They can contact me via my email, and then we can discuss how we can share the leachables predictive model. The thing is, we are currently in a phase that we have a master thesis here, where the student is elaborating a really huge model, a model with different aspects. My model was a very, very generic and very simple one with only six compartments, and he is now setting up a model with a huge number of compartments with different bags and tubes and connectors and all these things. And he is also going to calculate the leachables load in that model. And if somebody is interested in that, yes, of course, we can share our model or share our way to build this model.
Thank you very much. So that looks like all the questions that we have today. I would like to take this opportunity to thank our presenters, Ekta Mahajan and Dr. Armin Hauk, for sharing their knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com/webinars. Thank you very much, everyone.
Thank you, and thanks, Ekta, for organizing and giving us this opportunity.
Yeah, thank you also to everybody to listen to us, and yeah, bye-bye.
Thanks.